Hello guys, welcome back to another video of teaching NogOps. Today I'm going to be teaching NogOps about nucleic acid and transcription. This is part 2. In the first video we talked mostly about nucleic acids. This video we're going to talk about transcription, but as always we have to give NogOps her smart glasses, just because everybody knows, as I said before, glasses just make you smarter. And that's what she needs right now. So, transcription. Before we get into transcription, we're just going to talk a little bit about nucleic acids and how they carry the information. So, we're going to start with DNA. So, we have DNA. You can think of DNA as a recipe book, okay? This has all the possible proteins, and they are stored in a sequence of genes in the DNA, okay? Then we have mRNA, also known as messenger RNA, and this is basically the copy of the recipe. So DNA is the recipe, mRNA is the copy of the recipe, and it's specific for one protein. So we said that DNA has all possible proteins. mRNA is a copy for one specific protein. So you have a recipe book. This is a copy for one specific recipe. And then this recipe makes a protein, which is basically a sequence of amino acids that are created by ribosomes during a process of translation, which we will probably talk about in other videos. So an example of proteins are enzymes, hormones, cytoskeleton, membrane proteins. There's a lot of them. So, just a little recap. We have a DNA, which is has small genes, and one strand of DNA is used as a template to make complementary RNA. Then we have something called transcription factors, which are very important for transcription to begin. So this transcription factors, they have to be bound to a promoter, which is a DNA sequence before the gene to start the transcription. So, transcription factors. I will draw it out so it will look much easier. Don't you worry, NogOps. Here, let's bring her here so she can really be with us. Okay, so we got transcription factors. We also have a DNA helicase. So these are stuff that you need to know, NogOps, right now, and then we'll cover it in a second after. So a DNA helicase. This is an enzyme that unzips the double helix of the DNA. And by unzips, it means it breaks the hydrogen bonds between the complementary strands, and this allows the RNA polymerase to access the gene. So this is an enzyme. Alright, and then this is... We're not going to put anything for transcription factors. Okay, C. Then we have an RNA polymerase. So as I said before, the RNA polymerase, it accesses the gene. So after the DNA helicase unzips the double helix of the DNA and breaks those hydrogen bonds, the RNA polymerase accesses the gene. So the RNA polymerase is also an enzyme. And this enzyme is used to join RNA nucleotides to the covalent bonds. And then we have RNA transcript, which is the result of this. So, transcript. So this is an unmodified RNA strand that is created by the polymer. So it's really important to know that this is unmodified, because there's going to be a process after where we modify it. And that modified version is called mRNA. Okay, so we have transcription factors, which are they bound to the promoter and they basically start the transcription. This is the beginning. They start the transcription. And then we have a DNA helicase, which is an enzyme that unzips the double helix of the DNA by breaking the hydrogen bonds between the complementary strands, and this allows the RNA polymers to access the gene. So this gene is used to join the RNA nucleotides to the covalent bonds, which creates an RNA transcript. This is an unmodified RNA strand that is created by the RNA polymers. So this contains the instructions that are going to be later used in the translation at the ribosomes to make a protein. And then finally we end up with mRNA, which is a completed modified RNA instructions. And this leaves the nucleus and it is used by ribosomes to be translated into an amino acid sequence. So RNA transcript does not leave the nucleus until it becomes modified into mRNA. Alright, now let's get into the whole transcription process. So let's do this in blue. So transcription has 3 parts. Yeah, let's do transcription. And let's bring her down. Okay, so the first part of transcription is going to be initiation. So in initiation, this is when the DNA helicase breaks those hydrogen bonds and it unzips a strand. And it exposes the template strand. So hold on. Helicase break bonds. Let's just do that. Break bonds. So actually we're gonna try to draw it here so Nogops can really see and understand what we're talking about. So you have a complementary DNA strand that looks something like this. Okay, you got your nitrogenous bases here. Okay, and then the DNA helicase basically comes in and okay, this is the DNA helicase. We'll just put it here. And it basically opens this up. Okay, it opens it up